I'm Po-Ru Lo. Most of you might be used to thinking of me as being affiliated with Harvard School of Public Health, where I did a postdoc with Elkis Price. But I recently started a faculty position 5 minutes down the street from where I used to be. So now I'm at Harvard Medical School and affiliated with Brigham Women's Hospital. So that's me. I usually like to start my talks by figuring out who you are. So show of hands, who knows what a mosaic chromosomal alteration is? Sort of, okay. And who knows what long-range phasing is? Oh, okay, great. I'll just have to tell you about it. So the outline of my talk is I'll start by talking about haplotype phasing, which, as someone asked in the audience this morning, is a key algorithmic component to along the pipeline towards imputation that Brian talked about. And I'll be telling you specifically about a statistical method that I recently developed called Eagle, which uses a new technique called the positional Burrows-Wheeler transform. I'll unpack all of that terminology as I go. The second part of the talk will be about applying this phasing methodology to detect mosaic chromosomal alterations, which, based on the show of hands, all of that was gibberish, but I'll explain it in due course. So hopefully this is not gibberish. The basic piece of biology that you need to know to understand the first piece of this talk is humans have diploid genomes. We all have pairs of chromosomes. One chromosome in each pair is from your father. The other chromosome in each pair is from your mother. You probably all knew that already. But what you may or may not know is that the way in which we measure genetic data is typically we sort of collapse together the paternal and maternal contributions. And so in this toy example, I'm showing you a paternal haplotype. Haplotype just means a sequence of DNA that you get from one parent. And then in this toy example, a maternal haplotype in red, AT, GGA. Now, that's what actually exists in your cells, but when you go and genotype or sequence this person, what you'll get out of your genotyping machine or sequencing machine is just a conglomerate of the two haplotypes. So at the first position, since you got an A from both the father and the mother, you get an AA. In the second position, however, you have two different alleles from your two parents, and so all you know is you got a CT, but you don't know where the C came from and where the T came from. So that information is called phase information. Namely, phase information is which allele comes from which parent. And so what I've told you is that phase information is lost by most genotyping methodologies. In particular, if I just told you what you got out of your assay, and you tried to recover what the haplotypes were, you would have to guess randomly. And if you guessed randomly, maybe you would guess right in the first position, where you correctly guessed that the C belongs to the father, the T belongs to the mother. But then in this other heterozygous position, maybe you guessed wrong. And if you guessed wrong, then we call this a switch error. So those are the basic pieces of terminology that you need to know. If you really just guess, you would have a 50-50 chance of guessing right. And so what I'll be telling you about is statistical phasing methodology, which tries to use large reference panels and to recover this kind of information imperfectly, but pretty accurately. And in particular, these days with very large genetic data sets, such as UK Biobank, what may surprise you is that you can actually recover this information much better than 50-50. You can actually recover it maybe 1,000 in a row correctly. So that is my very brief intro into what statistical phasing is all about. Why is it useful? I can more or less skip these slides because Brian already told you why it's useful. It's useful for a lot of things, but in particular, one of them is for this paradigm called imputation, where you collect data, add a small subset of genetic markers, impute it using a reference panel to a much larger, ideally the whole sequence. And the key part of it that Brian talked about the imputation part this morning. The key other missing piece is that before you run imputation, you actually need to phase the data that you get from your genotype array. And that's roughly because when you want to impute using a set of reference haplotypes, what you essentially are doing is you're trying to match up chunks, haplotype chunks that were inherited identical by descent. And so basically, you want to start by figuring out what do your father's haplotypes look like, what do your mother's haplotypes look like, and then match them at a haplotype level to haplotypes in your reference panel. Okay, so how do we recover phase information? There's a basic principle of phasing, which I think is useful to develop intuition. And going back to our toy example, what I've now done on the left is I've just narrowed down to looking at the two heterozygous sites in the toy example. One position where the father had a C, mother had a T. Next position where the father had a C, mother had an A. And what we measured, what we got in our measurement was just CT and AC. And so we're trying to figure out whether the haplotypes that were actually transmitted from the mother and the father were CA and TC or CC and TA. And if the only information I gave you was the genotype calls for the individual in question, you would have no way of knowing what the right answer was. However, now suppose I tell you a little bit more information. Suppose I give you information about statistical correlations between nearby markers, also known as linkage disequilibrium. And in particular, suppose I tell you that based on a reference panel, whenever I observe a C in the first position, then at the next position, there's a 90% chance that I'll observe a C, whereas there's only 10% chance that I'll observe an A. And suppose you also know that if you observe a T at the first position, then there's a 20% chance of seeing a C at the next position and 80% chance of observing an A at the next position. Well, then, as probably most of you have already figured out, you would be able to infer that the correct set of haplotypes is much, much more likely to be C followed by C and T followed by A, as opposed to the opposite combination. So that's the basic principle, is to try to leverage statistical correlations in a larger data set. But we can do a little bit better. And the way that we can do a little bit better is to try to think about, well, I told you sort of observationally that there are correlations. There's LD, linkage disequilibrium, between nearby markers and the genome. But how did those actually arise? And the answer is they arose because DNA is inherited in blocks. You get chunks of chromosome, sometimes close to an entire chromosome from one parent at a time. And the only way that chromosomes are broken up is by recombination, which occurs relatively infrequently, at least on a generation by generation level. So if we can actually model this process that gives rise to these correlations, then we can do a better job of inferring haplotype structure and inferring haplotypes. So we've heard a lot already about hidden Markov models, and you've now seen a couple different renditions of this figure. What do I want to highlight about this part of the figure? Well, I guess I've mostly wanted to kind of remind you of, this is very similar to a figure that Joe showed you in his talk. And this is sort of the basic diploid hidden Markov model that people use to perform phase estimation. And the way that the hidden Markov model works is, again, you start with a reference panel of haplotypes. You imagine generating a new individual by copying chunks of haplotypes from the reference panel. So we're generating a new diploid sample. And so what this figure is showing is you start off just imagining maybe this paternal haplotype was inherited from some common ancestor of this blue individual. And you sort of walk along it for a while, copying, and then after a while, then you undergo a recombination, which means that you have to swap over to copying from some other haplotype. And so, okay, so this is sort of the basic model, and this Lee Stevens hidden Markov model does a really good job of incorporating kind of the key aspects of haplotype structure, namely the fact that haplotypes are inherited by descent, but also that recombinations occur relatively infrequently, and so that the jumping between copying states allows you to do that. And so this has been a tried and true approach. over the past 15 years, basically ever since the Lee and Stevens model came out. And there have been multiple generations of haplotype phasing methodology that have all, in one way or another, been building off this kind of hidden Markov model. Brian was being a little bit modest and not mentioning that he had done some of the foundational work with Beagle. And so one thing that you might wonder is given that I've just sort of summarized all of these 5 generations worth of phasing methodology, saying that they're all using hidden Markov models, well, what was different between each of them, and why was there something new to be doing every 3 or 4 years? And the answer is really that the data sets, the data sizes have been increasing dramatically. And I guess one difference from my talk versus Joe and Brian's is I don't have this nice, the sort of cost of sequencing going downhill. Basically, the idea is every couple of years, you wind up with a data set that's maybe 10 or 100 times bigger. And then all of a sudden, the approximations and the modeling that you were doing with your previous iteration of a hidden Markov model-based method don't become so good anymore, because now you have to be kind of moving from quadratic time to linear time to sublinear time. And so the sort of first sort of general point I want you to take away from this slide is that whereas hidden Markov models are generally very fast, they're polynomial time via dynamic programming algorithms like forward-backward, Baum-Welch, Viterbi. Because genomic data has been expanding so rapidly, we're starting to need even faster algorithms and starting to need to innovate on the way in which we use these models. And so that's what I'll be telling you about in the next couple of slides in terms of the method that I developed recently. And so this method is called Eagle, sort of a play on Beagle. It's now new-ish. We published it in 2016 in 2 papers, except that the second paper made the first paper obsolete before the first paper was published. So I'll only be telling you about the second paper. And there's sort of 2 key ideas that go into this version of a hidden Markov model-based statistical phasing method. And the first idea is kind of a departure from the way in which we usually think about hidden Markov models. So we usually think of hidden Markov models as a way in which you can model a generative process and perform some posterior inference by integrating over the whole space of combinatorial paths through your generative model. And that's what pretty much every hidden Markov model-based statistical phasing method has done up to this point, is you've tried to sort of approximate your state space maybe to make the computation feasible, but then use standard hidden Markov model algorithms to just integrate over the whole space and consider all possible paths. So the first key thing that we did differently was we decided to try to dispense with that and to try to instead of integrating over the whole space, just try to focus our computational attention on a small subset of the most likely paths through the space. So I'll try to give you a little bit of intuition behind this. If I told you right now, if I handed out a quiz where I gave you an individual sequence to phase and I gave you a bunch of reference haplotypes, what would you do? If you just had to do the best job that you could in the next 5 minutes? Well, you probably wouldn't start by building a hidden Markov model and sort of writing down a whole bunch of states and computing a dynamic programming table. Instead, what you would probably do is you would probably say, OK, well, I'm just going to start looking at pairwise correlations and look at the first marker and sort of look at what comes after the first marker and where the correlations are, like which position which haplotype phase is most likely for the next one, and then kind of walk from left to right across. And you probably wouldn't get it perfectly right. You probably wouldn't get it with optimal accuracy, but you would do a pretty decent job. And sort of hidden in that intuition is the idea that you kind of only care about a subset of paths through the hidden Markov model, because most of the paths that you might try to integrate over are just not very likely. Like in this toy example, you could probably say, OK, well, for the first and second positions, I'm pretty sure it's going to be CC and TA. And I can just sort of throw away the rest of the state space. And so that's kind of intuition. What the slide is trying to show you is that you can do a little bit better. If you wanted to be a little bit smarter and not just look at two consecutive positions at a time, you could start to sort of expand across from left to right. Like you could start to think about, okay, well, maybe at my first position, I've now encoded all of these alleles as zeros and ones. Maybe at the first position, assuming that you start here, you're most likely to see a 0. And then if you start with a 0, then what are you expecting to see next? Well, maybe you're more likely to see a 1 than a 0. And you can kind of walk across from left to right, and that way incorporate a little bit more history into the knowledge that you're trying to make use of as you do this phasing. So I've told you now that we can save a lot of computational time by sort of homing in on the paths that we want. And a good way to home in on the paths that we want is to have a model of haplotype structure that sort of starts at a given position and then sort of expands from left to right. There's a challenge, though, which is that because of the possibility of recombination, we have to sort of be able to do this starting from the first position, but also starting from the second or the third position or the fourth or the fifth position, because of the fact that these haplotypes that we're trying to copy, they sort of start and end randomly, or they seemingly randomly across the genome because of recombination. And so if we actually wanted to implement this procedure, naively, we would need to build one of these trees starting from each possible position in the genome, which would be expensive. And so that's where the second key idea behind Eagle comes in. And the second key idea is an efficient way to represent haplotype structure. So we know that we kind of want to represent the haplotype structure using these weighted trees, but it's sort of difficult to build them, or at least naively, it's sort of difficult to build them. So each of these trees is supposed to start from a position in the genome and extend all the way to the end of whatever section you're trying to phase. And so if I say that M is the number of markers that I'm trying to phase, the number of SNPs, and N is the number of samples that I'm, the number of, let's say, reference samples that I'm working with, then naively, it takes order mn time to build each one of these trees. And I need to build one starting from each position in the genome. So that means I'm expending order of m squared n time. And naively, it will also cost order of m squared n space to store all of these trees. So that seems like a non-starter. And when I started to work on this problem, I sort of assumed for a long time that there was just no way to get this approach to work. However, then I had the good fortune of going to the Probabilistic Modeling and Genomics conference and hearing Richard Durbin talk about the positional Burrows-Wheeler transform. And so the positional Burrows-Wheeler transform I'll tell you about in the next slide. But basically, the positional Burrows-Wheeler transform it enables an algorithm and a data structure that allows you to both generate and store all of these, the series of trees in only linear time. So every single tree, but only in linear time. And I'll attempt to tell you in a slide how that works. And fortunately, the positional Burrows-Wheeler transform, despite its very long name, is actually relatively easy to explain. You could think of it as a way in which you can sort suffixes of a batch of sequences. So in this example, on the top, what I'm showing you is a sort of a partial a snapshot of what the positional Burrows-Wheeler transform is doing. So the positional Burrows-Wheeler transform sort of works from right to left, and at each stage, at each position, it gives you a sort order of all of the sequences that you have in your data set. So at iteration m, in this top figure, what I've done is I've shown you that the positional Burrows-Wheeler transform has, sort of by definition, it has sorted all of the sequences that you have starting from position n and moving forward. So the first position is this 0, 1, 0, and then a 0, 1, 0, 1, and then a 0, 1, 1, 1, 1, 1, 0, 0, 1, 1, 0, 0. So these are all sorted. And the positional Borers-Wheeler transform is then tasked with, OK, at position m+1, in other words, when I incorporate a new position into my sequences, how do I resort all the sequences now that I've augmented them on the left? And the clever trick is that you can do this simply by taking all of the sequences that you augmented a 0 with, which in this case is the first one, and then the third one, and the fourth one, and the fifth one, and just moving them all to the front. And then taking all the sequences for which you augmented at 1 and moving them all to the end. And you can hopefully you can kind of see why this works. The reason is that you're kind of assuming that everything is sorted starting from position m. And so now that you've introduced a new position, as long as you move all of the zeros to the front, then they will remain sorted because everything after them is sorted. And then if you move all of the ones to the bottom, then they'll remain sorted. And also they'll all be after everything that starts with a 0. So is that clear? Nods. Cool. Yes?
Can you do this in sort of efficient pointer way so that you don't shift everything like info of any time?
Yeah. So it's all done through some basically pointers, storing pointers. And remarkably, you can actually code this in about 10 lines of code. And I'd been aware of this paper before Richard Durbin actually gave the talk that I went to, but I'd been too lazy to read it, sadly. And then when he gave the talk, then I realized, oh wow, it's actually incredibly simple. And so this is what Richard Durbin did with the PBWT. Well, this plus some compression stuff. But I actually don't need to use the compression stuff here. So the little piece of innovation that I did to add on to this PBWT was going back to building these data structures of weighted trees. So it turns out that, well, you can actually just sort of read that off directly from what you get out of the PBWT. So the PBWT, I just told you, it gives you a lexicographic sort on all suffixes starting from each position in the genome. And now those of you who have a computer science background probably know that a lexicographic sort is basically equivalent to a binary search tree. And so once you have a lexicographic sort, then you can basically just read off the tree, exactly the tree that you want. So that is,
yeah. Stupid question. Why do you go from right to left? Why are you sorting suffixes and not prefixes?
Oh,
Particular reason?
You could do it in either way. I guess I'm doing it from right to left because I wanted to preserve the intuition of sort of walking from left to right when I'm actually building these trees. So basically, if you walk from right to left, then you'll get sorted suffixes, and then you'll have trees. Those will correspond to trees that sort of go in the forward direction. Or you could do an analogous thing and walk from left to right, and you would get prefixes, but then your trees would sort of go in reverse direction. Yeah. All right. So that's all I'll say about the Eagle methodology. I have a couple slides to convince you that it works on real data. On the left side of this slide, these are plots of runtime of Eagle compared to the previous state-of-the-art ShapeIt, and it's typically about 20 times faster than ShapeIt. And then on the right side is an accuracy plot where the y-axis is mean switch error rate. Lower is better, and Eagle actually does slightly better than ShapeIt in terms of accuracy. When I've given this talk before, people have asked me, but wait, you just told me that you aren't integrating over the whole space. So how is it possible that you're more accurate? And the reason is that pretty much every hidden Markov model has had to make other approximations in order to keep the computation time down. So because of these advantages, the two imputation servers, Michigan and Sanger, actually put Eagle in production before we finished writing our manuscript, which was kind of cool. And there's been quite a bit of use of Eagle in the genetics community, TopMed and Million Veterans Program, GTEx, in the past couple of years. So at this point, I'm going to transition to the second piece of my talk, which is slightly more biological focused, but still with a heavy computational component. And this is going to be about using phasing to detect mosaic chromosomal alterations. And so I promised I would define that. So here are a pair of definitions on a slide. First, I'll define mosaic. For the purposes of this talk, what we mean by mosaic is just any mixture of normal and mutant cells that's caused by mutation followed by clonal expansion. And so you're probably used to thinking of your cells as all having the same DNA that you get from your mother and your father. But of course, during the course of your life, different cells can wind up with different somatic mutations in them. It might be caused by DNA damage. And most of the time, those somatic mutations don't really result in anything because the cell that picks up a somatic mutation eventually dies. But once in a while, a cell that picks up a somatic mutation might wind up proliferating. And the most typical example of this would be in cancer, where you basically start with somatic mutation, and you wind up kind of exploding into a big tumor. We can see this in less extreme situations, though, in the blood. So that's what mosaicism is. The second thing I need to define is what's a chromosomal alteration. And that is just any large-scale change in chromosome content of a cell. So in this cartoon on the bottom, I'm showing you a normal cell, which would have 2 copies of each chromosome, one maternal and one paternal. And then different things can happen somatically. You might wind up having a deletion of a chunk of a chromosome. You might wind up gaining a chromosome. Or you might actually wind up having this hard-to-pronounce term, a copy number neutral loss of heterozygosity, which basically means that a chunk of one chromosome, let's say the maternal chromosome, has replaced a chunk of the corresponding paternal chromosome. For the biologists in the audience, if there are any, that can happen through mitotic recombination. So there's a recombination that actually happens during mitosis. So that's what mosaic chromosomal alteration is, or, well, a mosaic chromosomal alteration is just a combination of the two things. So when you get one of these events in a cell, and then the cell starts to proliferate, such that it winds up, its clonal daughter cells wind up comprising a substantial fraction of the cells in your body. So to give you a little bit of motivation about why people care about this and what's known about it, there were a couple landmark papers in 2012 in Nature Genetics that showed that mosaicism, specifically these mosaic chromosomal alterations, can actually be detected in a few % of elderly individuals who don't have cancer. And that's kind of surprising because, well, you wouldn't really think that in your blood you're going to wind up with a whole lot of clonal mutant cells that are just floating around in your bloodstream. The other thing that they found in these papers was that even though the people in their studies were initially cancer-free, if it was found that somebody in the study had one of these events, then it increased the risk of a future blood cancer by a 10-fold factor. So that sort of suggests that, well, these events are interesting. In particular, that made us ask a couple questions. We were curious about whether we can actually improve the detection methodology to detect even more mosaic events that were sort of missed by previous methods. And it also made us wonder whether we could identify specific types of these events that particularly predispose to cancer. So that's kind of the biological motivation in a nutshell. Again, since this is a methods conference, though, I'll focus mostly on the methods from here. So how do we go about detecting these, and how do we improve the detection sensitivity? We can have a little bit of confidence that we can improve it because the previous papers were very honest about the fact that they were only telling you about what they could detect, suggesting that there's probably something else out there to be detected. And the basic intuition here is that the signature of all mosaic chromosomal alterations is that they create an allelic imbalance at heterozygous sites. So I'll unpack that a little bit. And in this cartoon on the left, what I'm showing you is an individual who has had a mosaic event, in this case, a copy number neutral loss of heterozygosity, where the maternal chromosome, or a chunk of the maternal chromosome, has replaced the corresponding chunk of the paternal chromosome. This has happened in a subset of the cells. And so now, if we think about how much maternal allelic content is there in this person's blood versus paternal allelic content, well, if we imagine sort of walking across the chromosome from left to right, then anywhere in the region that was not affected by the event, we would expect to see a 50-50 balance between the amount of maternal chromosomal content and paternal chromosomal content. However, in the chunk of the chromosome in which the event occurred, now what has happened is that some fraction of cells, the cells that were affected by the event, are now fully maternal and have lost their paternal contribution. And so if we just average across all of the cells that are in the blood sample, what we'll observe is that there's a shift such that the cells are now greater than 50% maternal and less than 50% paternal. So that's the basic signature that we're looking for in this data. The challenge, though, is that going back to the beginning of the talk, we basically never know which alleles are maternal and which alleles are paternal. All we get is this unphased data in which we can measure the amount of allelic intensity at any given SNP, but the readout that we get is something called a B allele frequency, or BAF. What does B allele mean? Well, it just means whatever Illumina decided was the B allele. There's an A allele and a B allele, and you can get the B allele frequency. Unfortunately, when you plot it, it doesn't really show you much. However, if you introduce phase information, then oftentimes you wind up with a much clearer signal. And the reason is because, well, let me just sort of tell you what you're seeing in this figure on the bottom. So on the top, I was showing you B allele frequency, or really the shift in B allele frequency away from 50-50. On the bottom, I'm showing you phased B allele frequency, where what I've done is instead of the B allele, now we're looking at haplotype 1 versus haplotype 2. What does haplotype 1 and haplotype 2 mean? Well, it's still not all that meaningful because it's just whatever came out of eagle. And it's not necessarily maternal or paternal. But what we do know is that on large chunks of the genome, haplotype 1 corresponds to maternal and haplotype 2 corresponds to paternal. And only every so often do we swap between haplotype 1 being maternal and haplotype 2 being paternal versus vice versa. And so what happens in the bottom plot is, well, we have an event which affects the right half of the chromosome. So on the left half, this phased B allele frequency is still sort of holding steady at 50-50. Then something interesting happens starting around 60 megabases, and you get a shift towards haplotype 2. Let's say haplotype 2 is on top and haplotype 1 is on bottom. And the shift remains steady until around 110 megabases, and then you suddenly swap the sign, and you have a shift in the opposite direction, and then you swap the sign again, and you have a shift in the positive direction. And so what's going on is that haplotype 1 maybe corresponded to the paternal haplotype, and haplotype 2 corresponded to the maternal haplotype across this whole chunk. But then there was a switch error, and the maternal versus paternal assignments were swapped, and so now you get this shift in the opposite direction. The good news is you still can obviously find a signal in here. And now what we have to do is, the task is, how do we detect such a signal in a manner that is robust to the fact that we're going to occasionally incur these switch errors that swap the sign? And so let me see, how much time do I have? Maybe about 10 to 15 minutes. So I'll tell you a little bit about the methodology of how we go about detecting these kinds of events. And in this slide, I've just sort of given you a little bit of just sort of a picture of a couple types of a couple examples of events that we want to detect, just to give you sort of the vocabulary of what we're looking for. The first example is an example of a copy number neutral loss of heterozygosity that starts on the P telomere. It incurred one switch error, and so you have this sort of shift to positive, then a sign flip, which is negative now, and then the rest of it is normal. And the second example, it's a chromosomal duplication, and so there's an event that spans the entire chromosome. Again, there are a few switch errors, such that you start negative, you switch over to being positive, then go negative for a long time, go positive again. And at the bottom, it's an example of a deletion that starts sort of randomly in the middle of a chromosome, goes along for a while, and then ends. So these are the types of events that we're looking for. How do we actually detect them? Well, the basic idea is to, once again, use a hidden Markov model. And that's sort of a natural thing to do because, well, kind of like for the same types of reasons that it was a good thing to do with phasing and imputation. Basically, the hidden Markov model allows you to account for the fact that you're sort of looking for shifts away from zero, but then every so often, but pretty rarely, you expect to see a sign flip. And so, what this hidden Markov model should do is basically it should have a couple different states. In this case, the state, each state corresponds to a possible shift away from the 50-50 balance. And in this example, I've shown you one possible way to do this is to discretize your possible set of shifts. So there's sort of a zero state would be no shift. The plus one would be sort of in some units of discretization, a slight shift, and then plus two would be a larger shift. And then what your hidden Markov model is supposed to do is it's supposed to reflect the fact that you can sort of walk through this model having an expectation of emitting roughly what the state label says you should be emitting, but then with some noise that's supposed to correspond to measurement error. And then the transitions in this hidden Markov model correspond to either phase switch errors, which flip the sign of the state that you're in, or to starting or ending an event, in which you would switch between a non-zero state and a zero state. Yes?
Looks like you have error bars in your delta path or something?
Yeah.
What are those from?
So those are actually because in this in this figure the raw data is really noisy, and so what I did is I just sort of aggregated data into bins, so that I think each of these points that were plotted is actually 64 SNPs that were just binned together.
Let me have this. So how do you get from the intensity from the bath thing into those?
So
How do you assign it to the haplotypes?
Oh, so
Allele?
yeah, so the bath just tells me what is the B allele intensity. And I guess we put that on a scale between 0 and 1. And then now all I do is I say, is the B allele haplotype 1 or haplotype 2?
Okay.
And that corresponds to a sign. And so I multiply in the sign, and then now I have phased bath. Yeah.
Well, Ruben, what about, do you have any problem with genotyping? I mean, so you've got these chromosomal alterations, it seems like that would delay traffic. With the genotypes you're getting, how do you work around that?
Yeah, so that's a good question. And so, in particular, if you're imagining these events at a very high cell fraction, like if an event has occurred in 50% of the cells in a blood sample, then what that's going to do is it's going to sort of totally throw off the typical genotype calling procedures because well, if you have an event that affects 50% of cells, then now your heterozygous SNPs, instead of being a 50-50 mixture of A allele and B allele, now they're supposed to be, I guess, a like 25-75 mixture of A allele and B allele. And so that will, in fact, wreak havoc on your genotype calling. In our application, though, we're actually mostly interested in sort of population scale detection of these types of events. And so the vast majority of events that we're looking for are at very low allelic fractions, like maybe 5% of cells or less. And so the cool thing here is that that's actually basically invisible to all standard QC procedures, and they just wouldn't notice such a small shift in the intensities. Yeah.
What's the advantage of using this method as opposed to just running averages? Absolute values?
Yeah.
Deviation from 0 to
Yeah. So there are a lot of ways in which you could do this, and one way would be sort of running averages or various segmentation type approaches. And really, the advantage here is it allows us to squeeze out a little bit more statistical power, because what we're looking for is less for events that we can call with 100% certainty. Those are the events that are sort of easy to detect. And we're really trying to squeeze out as much power as we can to pick up the events that are really quite hard to detect. And so I guess, for instance, on the previous picture, like all these events that I showed you, you don't need a hidden Markov model to detect. You can just eyeball it and use the eyeball test.
But how can you tell it's not false positive?
Yeah. So that's exactly what the hidden Markov model allows you to do. Is it sort of puts you on a statistical footing to be able to assign ultimately a p-value to an event being real. And now I'm almost out of time, so I guess I'll very briefly tell you some of the details of how that actually happens methodologically. So I showed you kind of a naive hidden Markov model that sort of would be the first thing that you might try to do. But in fact, it's actually kind of it gets more complicated than that pretty quickly. So naively, the most simple thing that you could try to do is actually implement precisely the hidden Markov model that I showed you in the previous slide and run a Viterbi algorithm to find a maximum likelihood path through the state space. But that actually runs into a couple of problems. And the first problem that it runs into is that the maximum likelihood path is only a single path. And in particular, in this type of scenario, it's actually the rule rather than the exception that you wind up with lots of different paths that have similar likelihoods. In particular, because of the fact that you have these rare phase switch events. So like a phase switch event you might expect every 1,000 heterozygous SNPs. However, you have a large number of SNPs, maybe 5,000 hets per chromosome, and so you expect to see about 5 phase switch errors. However, if you actually think about what that means for a hidden Markov model, for any path that goes through 5 phase switch errors, it's going to accumulate penalties of 0.001 each time. So you're going to get a probabilistic penalty of 10 to the minus 15 just for going through a typical number of phase switch errors. And that's kind of bad. And so to get around this, we actually need to think about how to integrate probabilities across more than one path. So there's a kind of nice way to do that, which is that a lesser known thing that you can do with hidden Markov models is beyond just trying to find the maximum likelihood path, you can actually integrate over the whole set of paths to compute the total likelihood across all paths through your hidden Markov model. And so what that allows you to do is, what you can do is you can sum over all possible paths, probabilities of all possible paths, and then compare that to the probability of just the null path, of no event. And then the inference is, if you sum over all possible paths and observe a much larger probability than the null path probability, then you can infer that there was probably an event, because some path must have had a moderately high weight, or rather some collection of paths must have had a high weight. So that's kind of the first thing that we can do to sort of tweak this. Yeah.
Why wouldn't just looking at the probability of observation over the most likely path and comparing that to the null path, why wouldn't that give you kind of the same signal?
Well, if you just take the most likely path, I guess the problem is that the most likely path might be one of several thousand paths that are almost the same.
That the probability for that particular path is tiny.
Uh-huh.
It would probably be the same thing for the null path too. But you can still compare them, right?
You could still compare them, but I think you would wind up in some cases finding out that your null path looked sort of better than it ought to have, because you're sort of not aggregating a whole bunch of non-null paths that sort of should be treated as one thing.
I see.
Yeah. Going back to Haki's question about how do you actually decide whether or not an event is real? There's actually a slightly fancier thing that you can do on top of this, which is you can instead of just sort of computing a maximum likelihood Viterbi path through your state space, you can create a family of hidden Markov models that's parameterized by a single parameter theta, which quantifies the amount of a deviation that you expect to observe. And in this family of hidden Markov models, you can compute the likelihood of observing the data under each possible value of theta. And for those of you who are familiar with this type of statistics, that actually gives you a chi-squared test statistic. And so that is kind of how we are able to decide with sort of more firm statistical footing whether or not an event is real.
You do that for a particular region, or you do it chromosome-wide?
We do it chromosome-wide, yeah. Yes?
So what's the ground truth here? Do you have samples where you cannot know what the fraction is?
Yeah, so there's sort of a question of when we benchmark this, how do we know whether we're actually getting correct results? And I guess there are a couple answers to that. So Well, one answer is we actually don't have ground truth. That's obviously not a very satisfying answer. But a better answer is we have examples that are kind of obviously real, where you can actually kind of look at the sort of you can eyeball these shifts, and so you kind of know that the shifts are real. But a better still answer is we can actually look at the age distribution of people who have these events, people who have events that are called and people who don't have an event called. And what you should expect to see is that people who have an event should be much older than people who don't have an event, just because these events tend to increase very rapidly as people age. And so what we can actually do is we can compare the age distribution of people with events, people with high confidence events versus people with low confidence events, and check whether that kind of matches up with what we expect to see based on false discovery rate thresholds. So I think I'm out of time. Is that right? So I'll skip through the rest of there's a couple other sort of statistical subtleties. I want to tell you in one slide about what we actually found. We analyzed the UK Biobank data set. At this time, we had the interim release, which was 150,000 samples. We were able to phase it at an accuracy of around 30 megabases between phase switches. And we ran the analysis, wound up with 8,342 events, which is about an order of magnitude more than previous papers. And I guess I have some biology slides, which I'll skip through. There's kind of a cool application to detecting early signs of cancer, because the UK Biobank dataset includes accruing cancer diagnoses. So the DNA was collected between 2006 and 2010, but then since then, cancer diagnoses have been accruing and being added to the dataset. And so we're actually able to detect early signs of chronic lymphocytic leukemia up to 6 years before the diagnoses actually happened. So with that, I should close this talk. Thank my collaborators, and and mention that I recently started a faculty position, so I'm now recruiting students and postdocs.